All right, let's get started. So, hello, I'm Regina Bauckham, the SSE president. It's a real pleasure for me to introduce this year's recipient of the Society of the Study of Evolution's Lifetime Achievement Award, Dr. Sam Scheiner. Sam has shaped evolutionary biology through foundational research, extraordinary mentorship, and decades of service to the field. As a researcher, he fundamentally shaped how many of us think about phenotypic plasticity, helping us understand how genetic and environmental factors interact to shape phenotypic variation and demonstrating that plasticity itself is a heritable trait that can evolve. For many of us, his 1993 review on the genetics and evolution of phenotypic plasticity was not just influential, but it was required reading. Sam's impact, however, reaches far beyond his scholarship. During 26 years at the National Science Foundation, he mentored and supported generations of evolutionary biologists, often in ways that shaped careers quietly but profoundly. And through decades of service to our societies, he has helped build the very structures that support our community, from education and outreach to student recognition. This award recognizes scientific contributions, mentorship, and service. Sam's career exemplifies all three: foundational research that changed how we think about adaptation, deep investment in the people and institutions of evolutionary biology, and a generosity of effort that has strengthened this field for decades. Please join me in congratulating Dr. Sam Scheiner. And with that, Sam, take it away.
Thank you for that very nice introduction. And I especially want to thank the Society for this great honor. I very much appreciate it. So today, uh, because this is lifetime achievement, I'm going to talk about research that I have done across my entire career. And for a lot of this, I am going to reach back to the work that I did as a graduate student for my PhD dissertation. I realized recently that that research had actually then been reflected very much in more recent, the empirical work, more recent theoretical work. So we'll be taking a look at all of that. But first, I just have to acknowledge the many different institutions that I served at over the years, the various funding sources. This is only a short list of the colleagues that have co-authored papers with me. I want to point out three people in particular: Jim Thierry, who was my graduate advisor for the empirical work that I'm going to be talking about today; my longtime collaborator, Bob Holt, for the theoretical work; and then my wife, Judy Scheiner, of course, for the many things supporting me over the many years, but also she was my field technician for much of the field work that I will be talking about. I also, because as was mentioned, I served at NSF, and part of this honor is for NSF. I have to acknowledge the many people who served in the program. It went through several name changes, starting out as population biology and physiological ecology, ending up as evolutionary processes, which unfortunately is no more. And these are the many people who served. Uh, so you should thank, if you know any of them, you should thank them directly. And I strongly encourage you to reach out to people who are still at NSF in various capacities, because they are dealing with a lot of difficult times right now and would appreciate the support. So, with all that said, let's get to the research. So, in many ways, much of my research over the years has really been uh, addressing this fundamental question: how do organisms adapt to heterogeneous environments, to variable environments? And in this case, heterogeneity is being defined from an organismal uh, uh, point of view, in which so environments are different if they result in different optimal phenotypes for a given organism and a given trait. Um, and The empirical story I'm going to talk about deals with this plant, the Anthonia spicata. It's a short-lived perennial bunch grass. Its common name is poverty grass because it grows in really lousy sites throughout the eastern United States. And the work that was done here at the University of Michigan Biological Station. So I was a graduate student at University of Chicago, but went up to UMBS, which is commonly called Bug Camp, for my summer research. And in particular, the work that I'm going to talk about took place on a set of experimental burn plots, which I'll show you in a moment, that had been established years earlier, beginning in 1932 by Frank Gates, who was the longtime faculty member there. The 1911 date you see here, that was not an actual experimental burn. That was actually the last time this whole area had burned naturally. But that area is where I used for my talk. And then the 1980 date, that burn was done for me as part of my dissertation. So the station very nicely decided that a new burn was due, and then I made use of it for my work. And just to show you what this looked like, so I was looking at Danthonia along a successional gradient, and here's what that gradient looked like. It starts out with a forest that gets cut and then burned. Following the burn, you've got aspen sprouting. Eventually, you get pine, white pine, red oak, and red maple coming in and filling that forest back in. So you have the successional gradient going from high light to low light. And Danthonia along that gradient. Here is data on that Danthonia. So you can see it starts out rare in the site. It increases in frequency. This is number of sample plots it appears in. It increases in frequency throughout to mid succession and then decreases to late succession, but it's still there at least 70 years after fire. And so the core question is, how does Danthonia manage to persist during succession? That was the question I asked for my dissertation. Uh, so stepping back a little bit and just asking in general, what are the various patterns and modes of adaptation to variable environments? As Pim Edelor and Dan Bolnick pointed out, you can look at that in terms of what changes within a generation, either in terms of a focal trait, which may not change at all. An organism may have a fixed development, or it may be plastic, it may vary, or the organism may vary its environment by either moving around through habitat choice or altering its environment through habitat construction. And then if that's what changes within a generation, then across a generation, you then have corresponding genetic changes. So if there's no change, that's equivalent to either having fixed mean trait values or variability, and I'll explain among individuals, and I'll explain what that means in a moment. Of course, plastic development means there's evolution of the reaction norm. Habitat choice is due to evolution of propensity to disperse or related behaviors, and then habitat construction, propensity to construct or, again, related behaviors. So from all of this, we end up with 6 different modes of adaptation: genetic differentiation, jack of all trades, phenotypic plasticity, developmental noise, habitat choice, and habitat construction. And again, I'll go through those in detail as we go through the talk in terms of what each of those means. My own career has been mentioned, has really focused on phenotypic plasticity as a mode of adaptation, how it evolves, the constraints on its evolution. But as I said, looking back on my career, and I've done theoretical work on all of these, um, I realized that the D'Anthonia work actually involved four of these six different modes of adaptation, and you'll see that as we go through the talk. So really the question then becomes, under what conditions is each mode favored? When do you get, differentiation versus plasticity versus habitat construction and so forth? Well, the answer is it's complicated. And we'll see that. So there are, besides actually talking about these different modes of adaptation, I also want to talk about how we study adaptation and or evolution in general, but in particular adaptation. So there's lots of different ways to study adaptation, and in my work, we have various sorts of empirical studies. So we have measures of natural populations. So here's the Anthonia growing in high and low light conditions. Um, Part of this work included reciprocal transplant gardens, so in nature, but manipulated to some extent. So this is the two gardens at the two ends of the gradient at the brand new burn and in the 1911 burn site. And then I also did greenhouse experiments in which I manipulated both light and water. So, what this gives us is a range of empirical approaches from natural populations to greenhouse or lab experiments. So, we have from one end more realism to more control. And although here it says transplant gardens, that really stands for the whole range of kinds of manipulations that people do in the fields, face rings, mesocosms, predator exclusions, all of those are sort of in between these two ends. But as you'll see, to really understand adaptation, you need to do sort of that whole range of studies. So the other way of studying adaptation is theoretical, and there's sort of two different modes. One is analytic models, and this is from one of my papers with Sergei Gavrilets. And I will say that if you ever see an equation in any of my papers, that is absolutely due to one of my co-authors. I am not a mathematician. So, I can understand the math, but I certainly didn't do any of the derivations. But on the other hand, I can code. The other way of doing theoretical work is through computer simulation models. And I've published a series of papers that are all based on this sort of basic model where we have 50 deans in a linear array where the optimal phenotype varies across that gradient, Gaussian fitness within. Critically, there is a life history that goes on here. So we start out with birth, development, movement, that is dispersal among deans, and then selection and reproduction, or uh, a second life history in which we have selection and then movement. So the key difference here is whether or not selection occurs in the environment in which development happened or in an alternative environment. And that clearly then uh, affects the predictability of the environment of development and the environment of selection. And you'll see why that is key. Uh, in terms of the genetics here, these models have two kinds of genes labeled non-plastic and plastic. Whether or not their expression is independent of the environment or environmentally dependent, depending on the simulation, 1 to 5 unlinked diploid loci. And so the phenotype here is simply the sum of the allelic values. As you can see, we have the sum of the non-plastic alleles and then the plastic alleles, where that's multiplied by some environmental factor. And so that's that's what allows alternative evolution of either genetic differentiation or plasticity. For other kinds of simulations, for those other modes of adaptation, I had simulations that involved uh, developmental genes, which caused random phenotypic variation, ones that affected likelihood of movement, and others that determined whether or not the organism would modify its environment. So, just like with on the empirical side, there's a range of approaches. The same thing is true on the theoretical side. So, on one end, you can think of sort of classic Haldane Wright genic models, usually limited to 1 to 3 loci because of analytic constraints, but they're analytically tractable, easy to interpret, and you can include lots of different kinds of genetic effects. At the other type of analytic model are quantitative genetic models, so Fisher and Landy models, which assume very many additive loci of small effect, also analytically tractable, and there's ease of interpretation, and here you can include many kinds of traits. And then between those are then computer simulations, which allow highly flexible genetic architectures. So you can include all kinds of different factors. You can include multiple traits. The nice thing about simulations, it allows exploration of lots of different scenarios that are not analytically tractable, and it allows for emergent properties and surprise. And you'll see one of those cases. On the other hand, sometimes they can be hard to interpret. So you could think of this again as sort of this gradient. On the one end, we've got complex genetics with simple phenotypes, or simple genetics with complex phenotypes, and then computer simulations in the middle, which can include all kinds of complexities. So, as I said, we have these 6 modes of adaptation. 2 of those modes involved fixed phenotypes, either genetic differentiation, right? We've got 3 different environments here, and then 3 different genotypes, each one that's fit to its environment. On the other hand, we can also have a fixed phenotype, a jack of all trades, where the individual is not optimal in any of those environments, but it's sort of partially fits each. So and if in the literature, these are often called specialists or generalists. Specialist is a reasonable term, but generalist is not, because there are actually lots of different kinds of generalists, which you'll see shortly. But when are these favored? Well, jack of all trades is favored when there is temporal variation. On the other hand, genetic differentiation is favored when there's spatial variation coupled with limited dispersal and or strong selection. And I'm not going to give you all the theoretical background, but these conclusions go back to models going all the way back into the 1950s. So this was sort of well known in the field when I was starting my work. And in terms of how that played out with Danthonia, so let's take a look at those transplant gardens. And in this case, so burn plot origin, you've got populations collected from along the environmental gradient from 1980, which is the high light, to 1911, which is low light. Total dry weight is a reasonable proxy for fitness in this case. And we have individuals growing in both gardens. And what you can see here is there's some genetic differentiation among those gardens. That is, the populations from earlier in succession do better in highlight than the ones from later in succession. But there's on the other hand, there's no evidence, no differences among any of the populations for growth under low light. And so this is sort of as you'll see, for a lot of this is sort of a partial modes. That is, in highlight, there is genetic differentiation, which tends towards being adaptive. But in low light, it's sort of a jack of all trades scenario where kind of everybody is sort of equally ill adapted to low light conditions. So those are the first two modes. Then we've got modes 3 and 4, which involve one genotype that can result in multiple phenotypes. And that's certainly what phenotypic plasticity is all about, where you've got this one genotype and an organism alters its development, its phenotype to match the environment that it is in. On the other hand, you've got what's been referred to as sometimes adaptive coin flipping or developmental noise. That is, a single individual gives rise to offspring which have variable phenotypes totally at random. And with the hope that if you don't know what the environment is going to be at the time of selection, that at least some of those individuals will uh, fit the selective environment. Uh, Again, in terms of how these are favored, so plasticity tends to be favored when the spatial or temporal variation is predictable. That is, when the environment at the time of development, the time that the phenotype is fixed, has a reasonable prediction of what then the environment of selection will be. And if not, if there's temporal or temporal plus spatial variation that is unpredictable, then that will favor developmental noise. And let me just show you some of that. But first, a little bit of a note about terminology. So this goes back to Bradshaw, which sort of helped define these terms of plasticity. So phenotypic plasticity is when a trait changes as a function of the environment. But then phenotypic flexibility is the ability of an individual to have high fitness in more than one environment. And I will say that these two concepts are often conflated or confused. So people talk about the phenotypic plasticity of the entire organism, and that's really not what it is, because it's really individual traits which are plastic, and then that can result in flexibility. But you can also be uh, flexible, have phenotypic flexibility by having a fixed phenotype that just happens to have high fitness everywhere. A second little note about terminology here, and that has to do with developmental noise, which is sometimes also referred to as bet hedging. You will see the following term in the literature. Well, and these two names, developmental noise is really about the mechanism or what's happening with the trait, while bet hedging is sort of the fitness outcome of that variation. And you will see bet hedging sometimes referred to in the literature as diversifying bet hedging. That is actually a redundant term. All bet hedging, which actually comes from economics and gambling, is by definition diversifying. That is, you're hedging your bet by betting on more than one outcome at a time, and it's then if people talk about diversifying bet hedging, they talk about conservative bet hedging, which is actually an oxymoron. And there's a perfectly good term for this, and that's called jack of all trades, which is actually much older. So we can contrast then developmental noise with jack of all trades. So what about those modeling outcomes? So these are the results of those computer simulations I talked about, and what happens when we have spatial variation. So in this case, we're varying dispersal rate along that gradient and looking at the relative plasticity at equilibrium for these two scenarios where individuals first move or first there's selection before movement. And in this case, optimal plasticity is when relative plasticity equals 1, and you've got genetic differentiation when it is at 0. And so as dispersal rate increases, you're getting increasing cross-generational variability. Increasing variability selects for plasticity, but if you move before selection, then even more movement means that there's increasing within generational uncertainty. And so you start then to select against plasticity and for genetic differentiation. So that's when you have spatial variation by itself. So in this case, predictable variation selects for plasticity. Uh, what about temporal variation? So in this case, in these scenarios, there's only a single Deem, which varies through time instead of that gradient. And for plasticity, again, we've got relative plasticity on the y-axis, and we've got two different aspects of variation: the amount of variation from one generation to the next, versus and the amount of autocorrelation in that variation from highly positive to negative. And again, you can see more variation selects for higher plasticity. There's a very slight effect of autocorrelation in this case. Now that's for plasticity. What about that other mode of adaptation, developmental noise? Well, again, developmental noise, or as it's labeled here, developmental instability. Again, as temporal variation increases, you increase selection for instability. And in this case, autocorrelation has an effect. So negative autocorrelation, again, you've got really big differences from one generation to the next. You select for that developmental noise because just who knows what the environment's going to be in the next generation. So temporal variation can select for either plasticity or developmental instability when each of them are the only option. So in these scenarios, either plasticity was an potential outcome or instability, but not both. What about the way that those two factors, spatial and temporal variation, might interact with each other? So now we go back to that plasticity model. In this case, this is with move first. Remember, under move first, we had under zero temporal variability. That is, if you look at sort of the left-hand side of this graph, you can kind of see that increase and then decrease in the amount of plasticity. And then as the amount of temporal variation increases, you select for plasticity, especially under very high dispersal rate and very high temporal variation. But if you notice, so those two things act synergistically. This is obviously a complex shape here. But the one thing you may have noticed is this little corner up here, right? High temporal variation, high dispersal rate. And if you follow around to the left-hand side of the graph, you will see that this is relative plasticity greater than 1. Now I said 1 was optimal plasticity. So what's going on here where we actually are getting more plasticity than we would expect as the in terms of matching the optimal gradient? Well, that is something that Bob and I termed hyperplasticity. That is when the reaction norm is more responsive to the environment than is optimal. So why would that happen? Well, think of it this way. We've got an environmental gradient with a trait value, and there's a mean phenotypic optimum along that gradient. But once you add in temporal variation so that that optimum is moving within any given environment, now you have an entire range of possible optima. And the optimal reaction norm then is then steeper than that mean phenotypic optimum. And so that's what's happening here in the case of hyperplasticity. So hyperplasticity is then favored when environments of grand offspring, because in this case phenotypes are fixed once per generation, when grand offspring have highly variable and unpredictable environments. And you know, we can talk about why it's grandchildren, but this is actually an alternative to developmental noise. So this is actually plasticity acting as if it was developmental noise, and we discovered that in this model because we did not limit the phenotypic range. In a lot of models of plasticity, they limit it to between 0 and 1, and one of the things that I like to do in my models is don't provide any limits and see what happens. And in fact, this popped out. This was totally unexpected. So that's the theoretical side. What about what's happening with Danthonia? So, first of all, let's look at what's happening in natural populations to natural selection. So, these are measures of selection on leaf length, uh, using two measures of fitness, fecundity, and mortality. And you can see that uh, in 0 years after fire, in other words, immediate in that highlight environment, there's positive selection for leaf length. But then uh, at uh, mid succession, when the canopy is starting to close in, we actually have stabilizing selection. So positive selection for longer leaves during early succession, stabilizing selection for longer leaves during mid succession. So we do have a gradient in selection on leaf length. And so what's actually happening, if we look at those transplant gardens then, and we look at the length of the leaves from those populations from along the gradient, you can see that in the open, populations from earlier highlight environments do tend to have longer leaves than those um, from late in succession. But interestingly enough, the 1911 population, so those are individuals that are planted back into the environment that they came from, they don't have longer leaves. So there's no evidence that there's any kind of adaptation at low light conditions. So we get genetic differentiation for longer leaves at high light, which is adaptive, but not in the reverse scenario in this case. And then we can ask, well, why is that? What kind of variation do we actually have in these populations for both genetic variation and plastic variation? The plastic variation being the variation due to the environment or genotype environment interaction effects, which was defined by this paper by Charles Goodnight and I. And what you see here, so these are variance estimates, and they are estimates. That's why you can get a negative estimated variance, because there's an error bar around that, is that there is high plastic variation in those early succession population, but at least on a statistical basis, there's no difference here in genetic variation. So plastic variation during early succession, but no differences in genetics or in late succession. So we do see this, we do see patterns of adaptation, but it's certainly not symmetrical along the gradient. So that was adaptation by Danthonia for plasticity. And you saw that, again, there's plastic adaptation for plasticity early in succession, but not late. What about other species? So this involves a meta-analysis of plant traits from 31 different reciprocal transplant experiments, over 181 traits. The nice thing about this data set is that almost none of these was designed to study plasticity per se, and so the traits do represent a reasonable random sample of traits. They were not picked specifically because they might be plastic. And what we found is that, in fact, the most common mode of adaptation in this case is genetic differentiation, and adaptive plasticity, which you can see here, is only about a third of the traits. So in general, adaptive plasticity is not the most common mode of adaptation. Okay, so that's plasticity, and I spent a lot of time on that because, as I said, I've spent a lot of my career looking at plasticity. What about those other modes which involve changing the environment, modes 5 and 6? So, habitat choice, as the name implies, an individual may be in one environment and it simply moves to an environment that matches its phenotype, or habitat construction, when an individual alters its environment so that it better matches its phenotype. And there are various, habitat construction, the paradigmatic example of that, of course, is beavers that build dams, alter wetlands to favor the kind of vegetation that they do well in, um, or birds building nests. There's various examples of habitat construction. Darwin looked at how earthworms affect soil, so this goes back to Darwin, and of course, habitat choice. Lots of organisms move around. Um, and so when are these favored? Well, choice tends to be favored when there's spatial variation and low costs of movement, while construction tends to be favored when the benefits of construction go to either the individual doing its construction or its near kin. So usually it's offspring. And that's been shown through a variety of models. Um, so what about Xanthonia? We don't think about plants as doing either choice or construction, but that's actually not what's going on with Xanthonia. So if you look at where the plants are growing in those natural populations along the gradient, you can see here that the plants in that mid-successional period, when the canopy is closing in, they are actually growing in places that are sunnier than they would be if they were simply growing in random locations. So, in a sense, they are choosing to grow in things that look like earlier succession. And that, of course, has a trade-off when it comes to soil moisture. In 1981, there was a 3-week period of no rain in the middle of the summer. These sites all dried out. But if you compare that top versus that bottom graph, you'll see that the plants are actually growing in drier conditions than at random because they're growing in sunnier conditions. So plants in general are growing in places that are brighter and drier than random. So there is some kind of habitat choice going on here in terms of where the plants are growing. And we can really see that here. So this is numbers of individuals of Xanthonia in that 1980 burn plot. In 1980, the burn happened in August, so there was some increased germination in the population prior to that burn. But then the following summer, Xanthonia is showing up all over the place, and this is all coming from the seed bank. There's a seed bank of Xanthonia seeds. So the fire stimulated germination, probably from the nutrients from the ash. And interestingly enough, though, if we then collect individuals from those various time points, which I did. So this is in July and August of 1980, both before and just after the fire, and then June and September of 81 during that period of germination. And this is now growth in the greenhouse under high and low light conditions. And as you can see, those individuals that are germinating after the fire are doing much better under high light conditions. So what you have is that these new individuals are adapted to high light. So there is habitat choice that plants do, and it's by where they choose in to anthropomorphize, choose to germinate. And so they germinate under high light conditions and are more adapted to high light. And on the other hand, I mentioned I also looked at water. There were no differences in any of these individuals with respect to adaptation to water. So although there's sort of plasticity with respect to light, there is not plasticity with respect to water. Water acts more sort of like a jack of all trades. So this is again where when you talk about plasticity or flexibility, you have to really be specific about what environments you're dealing with. So um, I talked about these various modes sort of one at a time, also through a bunch of different models. I looked at how they interact. Remember sort of that question of which modes are favored under which conditions, especially when you allow evolution, the potential for evolution of more than one mode. So in general, if there is environmental heterogeneity, then plasticity is favored over either differentiation or that jack of all trades. If you allow for both plasticity and noise, plasticity will win out. If you allow for both construction and choice, construction gets favored over choice. If you put in spatial variation with construction, then you get differentiation gets favored over plasticity. So that's with construction. With choice and spatial variation, then genetic differentiation is favored over either a jack of all trades or plasticity. On the other hand, once you add in temporal variation, then that flips the modes of adaptation so that plasticity gets favored over the jack of all trades or differentiation. So what this is saying is that what modes are favored is very complicated. It depends on a variety of possibilities and how they might interact with each other, with the caveat that the above, this is sort of a summary of the various modeling that I did. That's a gross simplification. You have to really look at the papers to see all the nuances that are going here. And more and different type of models are certainly needed to be explored. I've only explored a fraction of the possibilities. So one more note about terminology. I see this in the literature all the time. People will talk about adaptation versus plasticity as if plasticity itself was not an adaptation. What they really mean when they say that is genetic differentiation versus plasticity. And please be more specific about what you mean, because otherwise people think of plasticity as a non-evolving trait. And one of the things that I've been pushing throughout my career is that plasticity itself is an evolved trait, or can be an evolved trait, certainly. So going back to that question I asked in my dissertation, how does Xanthonia manage to persist during succession? Well, through a variety of ways. First of all, through habitat choice. Seeds germinate and plants grow in higher light environments than average, especially following fire. And those individuals are more adapted to a high light environment. But there's also genetic differentiation, with individuals from early in succession being more adapted to high light than those from late succession. So there's phenotypic plasticity, with plants in those high light environments having longer leaves, which is adaptive. On the other hand, all populations are able to grow and reproduce in low light, which is sort of a jack of all trades kind of adaptation. And with respect to water, which I didn't really talk about because there's nothing interesting there. There's just no differences. I manipulated water in the greenhouse and found no effects. So in this case, how does it persist? It happens through multiple modes of adaptation. It is not just any one single mode at a time. So, some takeaways. First of all, methodological. When you're doing your experiments, consider all possibilities, all of those possible modes. Use multiple empirical approaches, field and laboratory and greenhouse studies, both observational and experimental studies, and a variety of kind of genetics, both molecular and quantitative genetics. Quantitative genetics has sort of fallen out of fashion, and I want to emphasize if you want to study selection, you really, on traits, you need to actually measure those traits. And when it comes to modeling, use a variety of approaches, analytic and computer simulations. They have different strengths and weaknesses. Importantly, especially with those simulations, do not constrain your models. Allow for the unexpected to happen. Don't assume, limits on things that might happen, and explore the parameter space extensively. Usually, my papers, you're in what gets published is not the entire parameter space that I looked at, because you find things that aren't interesting, but then you may also find things that you did not expect. What about empirical? Well, when attempting to determine patterns and causes of adaptation, you really need to consider the totality of the circumstances. What's the grain of spatial and temporal variation relative to things like gene flow, life history, development, physiology? What kind of constraints or costs there might be on your traits? What modes of adaptation are possible or plausible? Think broadly and look at related species and what kind of modes of adaptation might exist there. So, uh, I've been studying plasticity for many, many years now, but there's still lots of open questions and about plasticity and beyond. So, what modes of adaptation are? How are they mostly driven by internal constraints or external patterns of selection? I've mostly looked at the latter, but the former can be very important. Um, are different modes associated with different kinds of genetic architectures, including epigenetics? We're now in a position where we can ask those kinds of questions. Why is adaptive plasticity and bet hedging, which I didn't talk about as much, tend to be rare? Why don't we see more adaptive plasticity? How much cryptic plasticity and cryptic choice is lurking out there? Are there phylogenetic patterns to modes of adaptation? Um, and then finally, to just finish off, because this is about my career, I just a little bit of advice to those in the audience. And you should think about your career as sort of an adaptation to temporal heterogeneity. You're going to see lots of variation throughout your career. So, one thing is, which mode are you going to choose? Will you be a specialist or will you be a generalist? And there are certain advantages and or to each of those. You may spend your entire career studying one species really, really well. Um, or you might be plastic, work on many different systems and questions. I've worked on Danthonia, Drosophila, Daphnia. Obviously, I have a thing about the letter D uh, in the things I try to work on. Uh, in terms of developmental noise, you can think of that as your students who might work on things totally unrelated to your own research. For example, my research had really nothing to do with what my advisor worked on. Um, uh, Habitat choice. Be prepared to change institutions and roles within institutions through time. And you can think of the development of collaborations as a way of constructing the habitat around you in terms of your research uh, environment. And so, with that, I will open this up to questions. And I cannot see anybody here. I will stop sharing. Well, no, I'll leave this up just for a little while. So if people have questions and I don't answer them now, or you think about things later, you can go to that email address. You can also, if you want to get any of those papers I talked about, go to Google Scholar and my webpage there, and you can download pretty much anything. So let me stop the share. Do we have questions? Okay. Distinguishing plasticity from signal-driven developmental changes that are part of growth or reproduction. So, yeah, so Rachel, that's a great question, and uh, I think what you're doing here is actually distinguishing outcome from mechanism, right? So signal-driven developmental changes, that is what adaptation by plasticity is, right? You're looking for some signal. It's going to then vary the phenotype in some way. And the question then becomes, what is the developmental time frame between when you receive that signal and when selection is going to occur, and is that development going to be fixed during your lifespan, or is it going to be variable? Um, the models, most of the modeling I've done has been with fixed development, uh, just just because it's for no particular reason. But yeah, there's you sort of are shifting the constraints. So the shorter the time between the between signal, uh, what's driving the signal, and the phenotype, then you're more and more driving the what controls adaptation to internal constraints. So physiology, a lot of physiology is sort of like instantaneous, right? And so in that case, it's those internal constraints on what your physiology can do that then determine whether how plastic that physiology will be, um, as opposed to like leaves in a plant, which in this case for the Anthonia, each year they're producing new leaves. But once they put out a leaf, that leaf has a fixed phenotype. So other questions? And I can't see hands. I hope somebody can.
Yeah, I don't see.
Colette. Hi, Colette. Go ahead.
Okay, I can't start my video, but I think you can hear me maybe.
Yes. Yeah,
Okay,
yeah, yeah.
I'm really curious about the fact that water has no effect in any of these studies with Danthonia. What do you
Oh,
make of that? Are there might there have been root phenotypes, for instance, that you're not measuring that are
uh,
plastic or
maybe. Well, I did look at root mass, and no, there are no differences in root mass, and they're grasses, so they have fibrous roots. No, I think it's just, I mean, Danthonia, I mean, as I said, it's called poverty oat grass because it grows in
yeah,
lousy conditions. So I think it's just, as a species, it's just adapted to grow under dry conditions. What I didn't say is these are glacial moraines, so they're very sandy soil, so they drain
yeah,
quickly. That's why groundwater went down really fast during that 3 week period in the summer. So in this case, there's just it there probably isn't selection to differentiate, right? It's
yeah,
just adapted to that condition.
yeah, I hear you. Okay. Thank you. Good to see you.
Yeah. Other questions? Oh, yeah, from Rebecca. Cost of plasticity, yes. Is the topic resolved? No. I mean, so certainly caught there. Well, there are costs and limitations on plasticity. Most of what I have looked at in terms of models that predictability in the environment, those deal with sort of these external limitations. Costs are internal, like a decrease in fitness, even if you're matching the optimal environment, is one sort of cost. And I mean, it's resolved in the sense that, yes, there certainly are costs. I mean, it's been shown, but uh, just like with selection and adaptation and fitness, it's sort of a trait by trait issue. So what you should do then, if you're studying plasticity, is then for the particular traits you're looking at, is there evidence of a cost of plasticity for that trait? It might be there, it might not. That was part of the work that I did on Daphnia, was actually then was measuring costs of plasticity, and yes, in fact, we did find it. So it's resolved in the sense that they certainly exist. I've included them in some of the models, but mostly costs are boring. You put in a cost and you select against plasticity, which can be important in some respects. For example, some models we've done on genetic assimilation, that's important. But I just haven't looked at it much from a theoretical perspective, but it's certainly important. And we have time for maybe one more question. Otherwise, people are going to want to start to jump to the various talks. At least in virtual space, we don't need to try to walk between sessions. So we do have time for maybe one more question if somebody out there has one. I don't see any. So as I say, feel free to uh, email me if you have any questions, or if I'll be hanging around the virtual meeting, and then I will be in Cleveland. So for those of you who can make it to Cleveland, I hope to see you there.